Good morning and welcome to another episode of Microbiology 101. Today, my fellow doctors and I will be discussing the poultry disease Mycoplasma gallisepticum. We hope you enjoy.
Mycoplasma gallisepticum is classified as a bacterium belonging to the class Molyssae and the family Mycoplasmataceae. The hosts are most poultry, including chickens, turkeys, ducks, and geese. The diagnosis can be confirmed by culture of Mycoplasma, PCR, and blood samples from live birds.
I'm Janae and doing the characteristical features of the organism. It has no cell wall, highly variable surface proteins, flash-shaped morphology. It has a unipolar terminal organelle, the bleb. It is the smallest self-replicating prokaryote, do not replicate in the environment. Most are host-specific, possesses a triple layer limiting membrane, but lack a cell wall. Do not stain well by Gram method. Highly pleomorphic, filtratable plastic form. Susceptible to desiccation and disinfectant. Most are facultative anaerobes, and cholesterol is required for growth.
Being a chronic respiratory disease, it is most often transmitted via direct contact and both vertically and horizontally. This can be between infected poultry's bodily secretions, such as feces, and susceptible poultry, which is very common in intensive poultry farming houses. The mycoplasma is also transmitted via infected aerosols, particles or water droplets in the air, or through the contamination of feed, water, and the poultry's environment. The bacteria can also be carried untransmitted to the poultry indirectly via humans on their fomites, such as equipment or clothing. Thus, good biosecurity is extremely important in preventing further transmission. It can be transmitted between birds of different species and in a transovarian manner, whereby infected hens transmit Mycoplasma gallisepticum to their eggs and thus progeny. It is contracted by the initial infection and colonization of Mycoplasma gallisepticum in the epithelium of the conjunctiva, nasal passages, sinuses, and trachea. In severe cases, the bronchi, air sacs, and lungs may also be infected. There is an incubation period of about 6 to 10 days. Once infected, the poultry may be a carrier of the disease for life, and thus excess stress may cause the disease to reoccur.
Mycoplasma gallisepticum is like all mycoplasmas and require an enriched growth media to survive. A variety of genera molecules as well as cholesterol is needed for them to grow. Mycoplasmas are dependent on their host cell for the essential nutrients that they can't produce and only grow on certain nutrients in the lab, making them fastidious. Mycoplasmas have the ability to survive outside the host, but not for prolonged periods of time. Some strains have been observed in a lab situation to survive up to 6 days. They die when they do not have the required nutrients to survive, meaning when these nutrients get depleted outside the body of the host, they can no longer survive.
I hope you have found this discussion on Mycoplasma gallisepticum informative thus far. I am Elizabeth Birchmore, and I'll be firstly focusing on the most common portal of entry of Mycoplasma gallisepticum into the bird. As it causes a respiratory disease, it is commonly inhaled into the respiratory tract via infected aerosols, and it can also enter transplacentally from the infected mother to the fetus within the egg. There are a few specific mechanisms that facilitate the exit route of Mycoplasma gallisepticum out of the bird. This includes the coughing of infected aerosols and the discharge of infected nasal mucus. Another mechanism includes the movement of infected mucus down the esophagus with the help of nasal cilia, peristalsis movement, and gravity till they reach the stomach, where some die, but others may reach the intestines via blood as well. This way, the mycoplasma can be excreted through the feces and therefore exit the bird.
My name is Taylor Rattray, and I will be discussing the types of samples you would take of M. gallisepticum to send to the laboratory, as well as the preservation and transport of these samples. Epithelia, such as the conjunctiva of the eye, nasal passages, and trachea, are highly susceptible to the infection by Mycoplasma gallisepticum. Therefore, swabs of these epithelial layers can be obtained and sent to the laboratory. The easiest site for swab collection would be from the conjunctiva of the eye, and this should be done by wiping a sterile swab over the area. It should be noted that the animal used for sample collection should be untreated so as to ensure the mycoplasma is present in large amounts for easy diagnosis. Mycoplasma gallisepticum can also be transferred within ova from infected breeders to their progeny, and this would allow for blood sample collection to confirm the diagnosis with M. gallisepticum with a hemagglutination inhibition test, as this organism would then be in the blood of the progeny. You should ensure that you have enough sample to send to the laboratory so that multiple tests can be performed. Attempts to culture Mycoplasma gallisepticum are optimized when the time from collection of the sample to direct inoculation of mycoplasma growth medium is as short as possible. The conjunctival or epithelial swab from the suspected infected host should be inoculated to mycoplasma transport media, and the sample should then immediately be stored on cold packs and transported to the Mycoplasma laboratory. The samples are warmed to 37 degrees C as soon as they arrive at the laboratory. Mycoplasmas are dependent on host cells for survival, which is why they need to be transported quickly. It should be noted that the sample needs to be kept moist, or the mycoplasma will die. This is why it is transported to a growth medium soon after being sampled. Keeping the swab itself moist, such as wetting with sterile water before sampling the tissue, would also allow better chance of survival of the mycoplasma.
Mycoplasma gallisepticum is classified under the domain bacteria. Other bacteria can either be classified under bacteria or archaea. Viruses cannot be categorized using taxonomic classification. Fungi is classified under the domain Eukarya. Mycoplasma gallisepticum has no cell walls because it cannot produce peptidoglycan. Other bacteria does have cell walls and differ in shape. Viruses is very small and complex. Fungi have unicellular or multicellular hive. Mycoplasma gallisepticum reproduce using budding or binary fusion. Other bacteria reproduce only by binary fusion, and viruses reproduce in their host cells, while fungi reproduce both sexually and asexually with spores. Mycoplasma gallisepticum exchange genetic material with horizontal gene transfer in hosts. Other bacteria exchange genetic material with transformation, transduction, and conjugation. Viruses also use horizontal gene transfer, and fungi exchange genetic material with hive fusion, sexual spores, and the fusion of nuclei during sporulation. Laboratory identification techniques of Mycoplasma gallisepticum includes culture on isolation, polymerase chain reactions, and serum plate agglutination. Other bacteria can be identified in the laboratory based on their gram status, their morphology and culture, the presence or absence of hemolysis, and biochemical tests. Viruses can only be identified using electron microscopes. Fungi can be identified in the laboratory by cultivation and the microscope examination of wet preparation.
Thank you for watching. We hope that you enjoyed that episode.
And protect your poultry.
And we hope that your poultry stay disease-free.